Hey guys, this is a whole topic summary for Edexcel, iGCSE, Organisms and Life processes. If you want to follow the video along, then you can go and download the free written guide from my website, which has all the bits that the examiners expect you to know in there, or Amazon will kindly print them. It's a bit of a beast off for you so that you can fill it in as you go along. Also from my website there are predicted papers for you so that you have loads and loads of practice when you're getting ready for the exam. Good luck guys. If there is anything else I can do to help you out, just leave me a message down below.
Life processes include movement, respiration, sensitivity, control, growth, reproduction, excretion, and nutrition. Now you'll notice that this is very similar to what you've learnt previously, so Mrs. Gren. But you'll also notice that Mrs. Gren suddenly has a first initial, control. Movement is when an organism can move around on its own, like deer through woods, or like amoeba through water, or like the flower of a plant following the sun. Respiration, organisms do respire. They change glucose to another substance, either with or without oxygen. Sensitivity, an organism can potentially sense what's around it and respond. For example, we're able to feel cold or warm, or when a pot plant falls over, it'll continue to grow upwards towards the light. Control, that's control of the internal environment, like a plant taking up more water in the heat or a person shivering in the cold. Growth, things get bigger. It's reality. From baby to adult, from seedling to tree, things increase in size. Reproduction, organisms generally tend to make other organisms, be it via sexual or asexual reproduction. Excretion, organisms release the substances that they do not need, for example feces, or carbon dioxide from humans, or even oxygen from plants. Nutrition, organisms need to feed on something. In animals, that's food. There are 5 general cell types. These include bacteria, animal, plant, fungi, and protoctist. Each of the 5 types of cells can be classed as either eukaryotic or prokaryotic, and the division generally lies over here. So the bacterial cells are classed as the prokaryotic cells, and the other cells, fungi, plant, protoctist, and animal, are classed as the eukaryotic cells. The reason these cells are classed as eukaryotic is because they have a nucleus, whereas bacterial cells have absolutely no nucleus, just a thread of DNA in the center of their cell. A plant cell has 8 parts that need labeling. So you've got your cell wall. Inside the cell wall, you've got the cell membrane. Inside of that, you've got the cytoplasm. You've got chloroplasts. You've got a vacuole. You've got a mitochondria. You've got a nucleus. And you've got ribosomes. Now each of these parts do something different within the cell, and they're each quite important. So if we start by looking at the nucleus contains the genetic information of the cell and codes for different proteins. The cell wall encases the cell, is responsible for support and protection, and also helps with turgidity of the cell. Cell membrane is generally classed as partially permeable and allows certain things to move in or out of the cell. The cytoplasm is the site of chemical reactions. The chloroplast contains chlorophyll, which is a green pigment, which assists the cell in photosynthesis and making its own food. The vacuole stores water and cell sap and is also responsible for turgidity. The mitochondria undergoes respiration to make energy, and the ribosomes are responsible for protein synthesis. Examples of plant cells that are quite important and you need to know them are corn, maize, cereals, and the legumes, peas and beans. This is an animal cell. On an animal cell, labels included are the nucleus, the mitochondria, the cytoplasm, the cell membrane, and the little dots here are ribosomes. These parts do exactly what the parts in a plant cell do. So the nucleus is obviously containing the DNA and the site of genetic material. Mitochondria is the site of respiration for energy. Cytoplasm is the site of all chemical reactions. The cell membrane allows substances into and out of the cells. And the ribosomes are there for protein synthesis. Examples of animal cells include obviously humans, but also things like the housefly and the mosquito. The animal and the plant cell can be compared with one another, and you need to be able to do this. You'll notice that the animal cell and the plant cell have slightly different parts. So the 5 parts of an animal cell are common with a plant cell, but there are 3 parts of the plant cell that the animal cell does not contain. That is the cellulose cell wall, the chloroplast, which makes the plant cell appear green, and the vacuole, which holds cell sap. Now, if ever asked to compare the animal cell with the plant cell and what differences they contain, those are the 3 that you look at. If you're asked to talk about similarities, it's the other items that are similar: nucleus, mitochondria, cytoplasm, cell membrane, and ribosomes. This is a bacterial cell. A bacterial cell has several labels. The first one is that it has a slime capsule. Underneath the slime capsule, it has a cell wall. Just like a plant cell, it has a cell membrane. And it has a cytoplasm. Now, unusually, a bacterial cell also has a few other features. It has a flagellum, most of them, some of them don't, but it has a flagellum to help it move. It also has plasmids, to contain DNA, and it has a chromosome loop, also containing DNA. Now, your slime capsule is obviously to do things like move between cells quite easily. Cytoplasm, it's for the usual things, site of chemical reactions. Cell wall, again, it's there for support, not as supportive as a cellulose cell wall. And cell membrane is to allow substances to move in and out of cells. Flagellum is to help with the movement. Now, you do need to know examples of bacterial cells, and it's really important that you can spell both your examples. So the first example is pneumococcus. That's spelt with a P. Pneumococcus. Coccus means round. So they're actually spherical little bacteria. They can appear individually or they can appear in groups. And your second example is your lactobacillus. Lactobacillus bulgaricus. That happens to be the bacteria that we use for making yogurt. And the lactobacillus, bacillus is rod-shaped. The lactobacillus actually has flagella all the way around and it's rod-shaped. You get three generalized shapes of bacteria. The three shapes of bacteria you get are the spherical shapes, the coccus bacteria, the bacillus bacteria, which are rod-shaped, and then you also get the spiral bacteria, which are slightly more unusual. And any type of bacteria can contain flagellum or not. Flagellum is not a deciding feature of a bacterial cell. What is a deciding feature as to whether a cell is bacterial or not is the chromosome loop in the center. It doesn't have a nucleus, it has a chromosome loop instead. Fungal cells are slightly different to the other types of cells, but contain a lot of the same features. They contain vacuole, cell wall, cytoplasm, cell membrane, nucleus. And sometimes they'll appear with mitochondria. Now the fungal cells, this particular one happens to be a yeast cell. Now yeast cells are the only unicellular fungal cell. That means they're a single-celled fungus, and they're used for things like making bread, beer, and wine, and sometimes in things like soy sauce as well. Now, yeast, although it's a single-celled fungus, it is the only single-celled fungus, and there are a couple of other fungi that you do need to know about. What's special about a yeast cell is that its cell wall is made up of a compound known as chitin, C-H-I-T-I-N. And chitin is slightly different from the cell wall of a plant. Plant cell wall is made up of cellulose. Fungal cell wall is made up of chitin. Mucor is a type of fungi. Specifically, it is a mold. We call the bushy bit of the mucor over here the mycelium. The mycelium is all of this bit. Now, each part of the mycelium has separate little bits. And each of these separate little bits are called hyphae. Now, this over here is an enlarged part of a hyphae. All right. Now, inside of this enlarged part of a hyphae, you'll notice that there are many nuclei. And that's because the cell walls of a hyphae has dissolved and they've become a single cell, so they have multiple nuclei inside of a hypha. You've also got your cell wall and your cell membrane, so the usual bits in a fungus, cell wall and a cell membrane. You've got your cytoplasm, and then you've got your vacuole. Now, when you're looking at the entire fruiting body of the mucor, the fruiting body is the whole thing. When you're looking at the fruiting body of a mucor, inside over here, the spores are found in the spore case, and generally your mucor is relatively small, so it'll be like the black misty mold that you see on bread, that kind of thing. So it's a small organism, but we class it as multicellular, even though there aren't separate cell walls between the hyphae. It's still a multicellular organism because of the multiple nuclei. The mucor feeds by something called extracellular digestion. Extracellular digestion is when enzymes are secreted from the cell. So they would be secreted into the surroundings. The enzymes would then digest the dead material in the surroundings, the dead organic matter, and the digested dead organic matter, the nutrients from it would be absorbed again by the fungus, and the fungus would then continue to grow. So you'll note that Nucor grows on dead organic material, as does most other multicellular funguses. You'll find mushrooms and things on a forest floor where the leaves are decomposing. You'll find molds on breads that have gone stale. The category of protoctists is a category of exclusion. What we mean by that is that they are single-celled, all of them, just like bacteria, but they have a nucleus. So you'll notice the nucleus, the large black spot in the middle here. There is a nucleus inside of protoctists. Now, if the cell you're looking at doesn't really fit well into all the other categories, it's probably a protoctist. Take this cell, for example. This is a sketch of Euglena, and Euglena is a protoctist. You'll notice that it has mitochondria. It has a cell wall. Right? It has a nucleus, but it's also got chloroplasts, fitting nicely into a plant category there, but right over here you'll see a flagellum. Now, flagella are generally only found in bacterial cells, but this is not a bacteria because it has chloroplasts. So, you've got chloroplasts that are only found in plant cells, you've got flagella, so it can't be a plant cell, it's not a bacteria because it's got chloroplasts. So, it's a protoctist, as it doesn't fit neatly into any of the other categories. Now, protoctists are like that. When you're looking at a diagram, you'll think, this is what's in a bacterial cell, this is what's in a fungal cell, this is what's in an animal cell, this is what's in a plant cell. This cell, a protoctist, doesn't fit neatly into any category. Here are some other examples of protoctists. In the top corner, we've got something called a paramecium. In the bottom here, we've got a chlorella. And in the top right, we've got something called an amoeba. Right, now I'll take you on a quick tour of these organisms. Now the paramecium, it's got its nucleus over here, so that you know that it's not a bacterial cell. Chlorella as well, and amoeba as well. They've each got a nucleus, so you know that they're not bacterial, but they're single-celled organisms. Now, paramecium, where it becomes confusing is, it has a cell membrane with flagella, and it also has temporary vacuoles, which is a characteristic of only a plant cell. Your flagella are characteristics of only bacterial cells, but because it's got a nucleus, it's a paramecium, it's a protoctist. Now your chlorella, you've got your nucleus, and you've got your single cell membrane around it. That's interesting. Inside of it, it's got chloroplasts. Now chloroplasts and a single cell membrane don't generally go together, and it's single celled, it's self-sufficient, so you're looking at a protoctist. Your amoeba has a nucleus, and it has an unusual shape with little podocytes on the end here. Podocyte, false feet. Now, the podocytes, interestingly enough, inside of the amoeba, you've also got contractile vacuoles. Vacuoles are not normally things found without a cell wall, because a vacuole will be found in a plant cell, which has a cell wall. So again, the amoeba, you're looking at something that doesn't fit into any other category. And the category of protoctists is the category of exclusion. Once you've found that they don't belong to any other category, you can decide that they are protoctists. This is a virus particle. The virus particle has a couple of main parts. In the center of the virus particle, you've got a DNA or RNA strand. It's never both, it's one or the other. Surrounding the strand, you've got a protein coat. Now that protein coat is very helpful because it means that your body can't detect the virus as a foreign being. Now some viruses will be quite cheeky, and upon destroying a host cell when it reproduces inside of the host, so it uses a host to reproduce, it will borrow the host cell membrane. And the cell membrane from the host will basically make it look like it belongs inside of the host's body. so it doesn't appear as a foreign object, which means that it doesn't get destroyed as a foreign object. Now, virus examples you need to know are the tobacco mosaic virus, TMV. Tobacco mosaic virus, needless to say, only comes up inside of tobacco plants. So no other plant, not humans, just tobacco only. You've also got HIV. You've also got flu. You've got a cold virus. You've got measles, mumps, rubella, and you've got polio. You only need to know 3 or 4 of them, but there are quite a few of them that are quite common. Now, viruses are not classified as cells themselves because they need a host to reproduce. They do not reproduce without a host, and they do not perform any of the other life processes like Mrs. Gren, movement, respiration, sensitivity, control of internal environment, growth, reproduction, excretion, nutrition. And although they do reproduce, they need a host to be able to do this. And they can't reproduce without a host, so they don't count as living. Pathogens can be any disease-causing organism. So the definition of pathogen is a disease-causing microorganism. Now, disease-causing microorganisms can appear in many forms. They can be bacteria, like your Salmonella bacteria or your pneumococcus bacteria causing pneumonia. They can be viruses, like measles, mumps, rubella, polio, the common cold, HIV, flu, etc. They could be fungi, like your athlete's foot or aspergillosis or any of those, ringworm even. Or they can be protoctist. Now, protoctists that cause diseases are plasmodium, like malaria. Malaria is a plasmodium, but it's also categorized under protoctists, a single-celled pathogen. So your 4 different kingdoms that can contain disease-causing microorganisms, that can contain pathogens, are bacteria, virus, fungi, and protoctists. There are various levels of organization in the body. They start off as simple as an organelle. Here we have a mitochondrion. They move on to cells, tissues, organs, organ systems, and the whole organism. So in this case, you'd have a mitochondria, which is for energy. It would house itself inside of a muscle cell. The muscle cells would make muscle tissue. That muscle tissue would be something like a heart organ. Then you'd have the circulatory system, which is an organ system, and then you'd have the full organism. The definitions of these are quite simple. So an organelle is the most basic unit within a cell. A cell is the most basic singular unit of life. And then the interesting thing is the rest of the definitions can be basically done with this little shortcut. So a something is a group of somethings that work together to perform a particular function. Okay, so we'll walk you through it. So, for example, a tissue is a group of cells that work together to perform a particular function. An organ is a group of tissues that work together to perform a particular function. An organ system is a group of organs that work together to perform a particular function. An organism is a group of organ systems that work together to perform a particular function. It's quite simple to do the definitions of each of the levels of organization because they're pretty much the same as the one before. The only two you really need to learn are organelle and cells. Inside of organisms, enzymes are catalysts. That means that enzymes speed up or slow down reactions. Now, certain enzymes are substrate specific. In fact, all enzymes are substrate specific. So an enzyme will only catalyze a reaction if it matches a particular substrate, right? So substrates need to fit into the enzymes. So here we have an enzyme. Here we have the part where the substrate fits, and that's called the active site. And now each of these enzymes have a substrate inside of them. Now the substrates are particular to the enzyme. I would not be able to have this substrate over here fit into this active site or this active site. Their shapes are extremely different. So this substrate would only fit into the active site of this enzyme. Now what would happen in the lock and key hypothesis is that you'd have an enzyme that breaks down a substrate, something like this, right? So it'll cause a division between the substrate, and then it'll cause the substrate particles to part, so into two different pieces. So this is what we would call products. So the products would then leave the active site and they'd continue to wherever they were needed. Now that is the action of an active site. Once an enzyme has catalyzed a reaction, so this enzyme over here, once the reaction is catalyzed, it'll just move on to the next substrate. So another substrate just like this would enter the active site. Its two parts would be broken up into products, and then the cycle would continue. Enzymes will be affected by a number of factors. One of those factors that would affect enzymes is pH. The other is temperature. Now, if the optimal temperature or the optimal pH is not utilized when an enzyme is trying to work, if the temperature is too low, the enzyme will slow down. If the temperature is too high, the enzyme will denature. Now, denaturing, just over here, is when the active site changes shape. So it wouldn't fit any of the possible substrates around. The enzyme's active site has changed shape in such a way that the enzyme is completely useless. This could also happen if the enzyme's pH was too high or too low. So the pH of the substance the enzyme was working in is too high or too low. There are graphs that depict the functioning of enzymes. So generally you'd have your rate of reaction up the side, and then you'd have temperature along the bottom, or you'd have pH along the bottom. Now, in a rate of reaction versus temperature graph, you'll notice that the rate of reaction steadily increases, gets to an optimal temperature, and drops off quite quickly. So the peak of this graph is what is known as your optimum temperature. Now the drop happens quite quickly, so the drop would be before 60 degrees Celsius. Your optimal temperature in enzymes that are operational in the human body, for example, are around 37 degrees Celsius in humans. Alright, now if you've got your rate of reaction versus pH graph, that is a generalized bell curve. Now the pH that enzyme would be most optimal at would depend on the enzyme. So for example, if we were looking at amylase in the mouth, that point just over there would sit at pH 7. If we were looking at pepsin in the stomach's quite acidic, so the optimal point of pepsin would sit at pH 2 or 3. If you were looking at the small intestine with enzymes like trypsin, its pH would be close to the optimal of 8. So the actual optimal point, the peak of the graph for pH, depends on which enzyme and where the enzyme is in the human body. Quite importantly, in your rate of reaction temperature graph, if it is over the optimal temperature, you're going to find that your enzyme denatures. Exactly the same happens on the pH graph. If the pH is too low for the enzyme or too high for the enzyme itself will denature. Respiration happens inside of the mitochondria of a cell. The mitochondria is the powerhouse of a cell, and it creates energy in the form of ATP. Now, the general formula for respiration is glucose and oxygen gives you carbon dioxide and water. Now, if you translated that into symbols, glucose is C6H12O6, oxygen is O2, carbon dioxide is CO2, and water is H2O. Now, your challenge is balancing it. It's really important to follow the balancing rule. The molecule without a 6 in it gets a 6 in front of it. So you'll notice that oxygen doesn't have a 6 in it, glucose does, carbon dioxide doesn't have a 6 in it, and water doesn't have a 6 in it. So the molecule without a 6 in it gets a 6 in front of it. Now you've got glucose plus oxygen giving you carbon dioxide and water, and that creates ATP. ATP stands for adenosine triphosphate. And adenosine triphosphate is like a basic unit of energy. What we have here is what we call aerobic respiration. Aerobic respiration happens in the presence of oxygen, but sometimes oxygen is not always present in respiration. Sometimes it can't be. And that's when you get anaerobic respiration. So aerobic respiration happens in all organisms when they can get to oxygen. Anaerobic respiration happens in organisms that are deprived of oxygen, that can't get to oxygen. Organisms that are deprived of oxygen undergo anaerobic respiration. It happens in humans in muscle cells, and it looks like this. You got glucose, and it converts directly into lactic acid. Now, lactic acid is a toxin. Lactic acid is a toxin that builds up and causes cramp. But it generally happens in people who can't get to oxygen, like, for example, the last couple of meters of a sprint, and you're running and you just don't have time to breathe because you've just put your all into getting to the finish line. Now, when there's no oxygen, lactic acid is created. Some ATP happens as well, but not as much as aerobic respiration, a little bit of ATP. Now, lactic acid can't be left in the muscle because it causes cramps and it's toxic. It's a really dangerous chemical. So what happens when you have lactic acid is you end up having an oxygen debt. Now, an oxygen debt is the amount of oxygen needed to break down the lactic acid. And the oxygen needed to break down the lactic acid basically gets added to the lactic acid. So you start with the lactic acid. At the finish line, just picture it. You've just finished a race. What are you standing doing? You're standing there going, you're panting, right? And that's to get the oxygen you need to make sure that all the lactic acid is broken down. So that's your oxygen debt, getting the oxygen you need to break down the lactic acid into carbon dioxide and water, two completely harmless substances, pretty much the same as having oxygen in the first place, but you'll get a little ATP there as well. Okay, so it helps break down the lactic acid into carbon dioxide and water once you fulfill the oxygen debt. Now that is only one form of anaerobic respiration. This is the type of anaerobic respiration that generally happens in humans. Right, there's another type of anaerobic respiration that can be found in yeast. The type of anaerobic respiration that can be found in yeast involves glucose breaking down directly into ethanol, which is an alcohol, and carbon dioxide. Now this happens generally in yeast when it is used to make beer, bread, and wine. Now I know what you're thinking. There is no alcohol in bread, and that's because of the high temperatures in the oven. When you bake the bread, the alcohol evaporates. Beer and wine is generally drunk cold, which means that the alcohol is not evaporated. So you've got the glucose, which then turns into ethanol and carbon dioxide. The ethanol is alcohol, and carbon dioxide, just picture it. It makes bread rise. It forms the pockets inside of bread, the little spongy pockets inside of bread, and it also makes the bubbles in beer. This type of aerobic respiration happens only in yeast. And what was yeast? It was a single-celled fungus. The definition of diffusion is when particles move from a high concentration to a low concentration down a concentration gradient through a partially permeable membrane. Now this happens passively. What substances move by diffusion? You're looking at things like gases, dyes, and liquids. What factors affect diffusion? If you look at factors that affect diffusion, there are things like temperature, concentration gradient, and also concentration of substances. How does diffusion work? And here's a quick illustration for you. So on the left hand side you'll see that I've got 6 large green sugar molecules and I've got 3 small blue water molecules. On the right hand side you'll see I've got 3 large sugar molecules and 6 small water molecules. Now in order for these two to even itself out, you'll notice that the water molecules are small enough to move through the gap in the partially permeable membrane, but the sugar molecules are not. So what happens is the water molecules will move across the gap and they'll dilute the sugar molecules so that both concentrations on both sides are more or less equal. The definition of osmosis is when water moves from a high concentration to a low concentration down a concentration gradient through a partially permeable membrane. Now the only difference between diffusion and osmosis is that osmosis happens with water. So basically osmosis is the diffusion of water. So what substances move by osmosis? Literally only water. Water is the only answer to that question. Osmosis happens when water moves from places like the soil into the root hair and then up the plant all the way to the leaves. Osmosis also happens when water moves from the blood into the cells or from the cells into the blood. So it's the movement of water. The movement of water also depends on other substances like sugars. Active transport is when sugars or other particles like minerals move from a low concentration to a high concentration, up a concentration gradient through a partially permeable membrane using energy. So active transport will only happen if energy is present. What substances move by active transport? You're looking at things like sodium, you're looking at potassium, you're also looking at sugars, you're looking at phosphates, those kind of substances. Now, it's very important to note that gases and water do not move by active transport. The reason active transport is needed is because it takes particles that are in high concentration anyway on the other side of a membrane and allows them to move that way. So if you have a partially permeable membrane and we have a high concentration of it, you'll notice that these molecules are too big to fit through the membrane. But because of active transport, the energy is used to move them across the membrane. So let's give you a generalized example. This is the blood, and this is the small intestine, where food is digested. Food particles, or building blocks like amino acids and glucose, will move across the partially permeable membrane by active transport. The energy is required to squeeze them through those small holes that they wouldn't ordinarily fit through. That is the end of all of the doubles content. So if you're taking only paper 1 or doubles, you can stop here. If you are taking paper 2 and are a triple student, listen on because the rest of your content is coming up. As an embryo grows, cells become specialized to carry out different roles in the body. We start with a sperm and an egg. That cell forms a zygote, and then you get cell division. So you get a first division, a second division, a third division, etc. And those in the third division can become specialized, and this, when the third division cells over there get specialized, it is called cell differentiation. Differentiation means that they change to perform a different function. The functions that specialized cells can perform are related almost entirely to their structure. This is a nerve cell, and what it does, because of its shape, is reach from the very tip of your spinal cord all the way to the tips of your fingers to be able to feel or to be able to detect stimuli. Here you've got a palisade cell, and a palisade cell is in a plant cell to be able to photosynthesize. This is ciliated epithelial tissue. This is a ciliated epithelial cell. And ciliated epithelium basically catches the dust and dirt inside of your nose so that you don't gather up dirt inside of your lungs. This is a muscle cell. This is a sperm cell. This is a white blood cell. This is a red blood cell. And this is xylem, which is found inside of plants. For cells to function differently or grow into different shapes, genes are switched on or off to make different proteins. So for a cell to have a different shape to another cell, a certain protein or a certain gene must be turned on or turned off to change the shape of a cell. A stem cell is a cell that is able to divide many times while remaining undifferentiated. You get two main types of stem cells. The first type is an embryonic stem cell, and they are found at the embryo stage and they differentiate into any cell type. The second type is an adult stem cell. Now this is generally found after 10 years old in most adults, and they can't differentiate into all cell types, but they can form cells like skin, so different skin cells. When you injure yourself, skin closes up. Bone marrow, and that makes white and red blood cells. It can form stomach and intestine lining. So when the stomach acid gets to your cells and breaks them down, it can reform stomach lining. It can also form nerve cells, and it can also form muscle cells. Now, stem cell therapy is something that is going on in the world of stem cells at the moment. Scientists are currently working with embryonic stem cells that they get from fertility clinics. So the embryos that are created that are not implanted into parents who can't have children are then used for a little bit of experimentation, and that is utilized to see if stem cells can be used to treat diseases such as Parkinson's disease. Can they use embryonic stem cells to grow nerve cells to treat Parkinson's disease? Can they utilize bone marrow stem cells to help leukemia patients produce blood cells that they wouldn't ordinarily be able to produce? Now, there is an ethical debate behind this. Some people object to embryos being used for medical purposes, despite the fact that they can be used to cure various diseases. There is a practical for this triples topic. The practical is where you change different pHs and see how they affect enzyme action. This practical can be found in the practical videos.
Ouch. Love you too, Crim.